Thank you, Alistair, and I'd like to reiterate that welcome. We are going to be looking tonight at the topic of gene reading. That's probably not quite the title that I had, and to really delve down on that a little bit more, we're going to be looking at reading your genes to personalise treatment, the future of cancer therapy. So, to really understand what I'm going to be talking about, we're going to need to take a few steps back. So many of you, at a quick glance around the audience, are a bit like me and left school a long time ago. So there are things about your genes that you might have forgotten. Now some of us, like me, had to relearn it, but I'm going to give you what I'm going to call I'll just work out which computer I'm using, here we go, a Genetics 101. So we're going to take a step back in time for a little while and talk about genetics and what DNA is, what your basic building blocks of your body are. We're going to then take a little bit of a look at what's normal and what's cancer. I need to then look at classic chemotherapy, the sort of stuff that many of you would be aware of, the degree of separation in this room. In other words, people who know somebody who has cancer or indeed have cancer themselves would be 1 degree. In other words, I would assume on basic statistics that everyone in this room knows somebody who has been affected by cancer. So many of you would know the word chemotherapy, but many of you may actually not understand a lot about what chemotherapy is. I then want to move into the new era, and this will be relatively briefly, but looking at genomics to target cancer. What's happened since we sequenced the human genome? That was only a short while ago. But really what we've seen is an amazing change in the way that we diagnose patients and the way that we will target cancer and other diseases, as a matter of fact. And really, as Alistair alluded, most of my work has been done in leukemia. So obviously I'm going to take examples from leukemia. And at the end to touch on the emerging face of clinical trials. So I do want to take also, this can be an incredibly sombre topic, so I kind of want to take a little bit of a light-hearted view to this as well and sort of break it up a little bit on the way through so that we don't get too morbid and we can, you can really follow the journey. So let's start with Genetics 101. What do we know about DNA and DNA replication? So there are about 20 to 30,000 genes in the human body. Now most of that is contained within the nucleus. 99.9% is contained within the nucleus of your cells. These genes encode proteins. These proteins are what make your eyes blue and your hair brown, or whatever colour it happens to be. It's really your genetic code. They do a lot of other things, clearly, apart from colour your hair and your eyes. This was a relatively new discovery. So there's some controversy even today about who really developed or understood the structure of DNA. Watson and Crick are probably names that most of you would know associated with the identification of the structure of DNA. But clearly there were 2 other players in this, Wilkins and Rosalind Franklin. And this was really first written up in 1953. So in 1953, as I said, a relatively short time ago, we understood at least the basic structure of human DNA. This group were given a Nobel Prize in 1962 for this discovery. This was a hallmark discovery. It has completely changed the way that we do medicine and the way that we do science. Now, as a woman in science, and as an NHMRC member of an NHMRC women's panel in science, I need to make a point here that Rosalind Franklin, who's clearly a female, was actually excluded from that Nobel Prize because they could only have 3. And so she was the 4th and she got dropped off. So she would have been one of the first women actually to win a Nobel Prize, but was actually excluded. She unfortunately, but maybe this is a linked thing, she actually died at the age of 37 of cancer. So she actually wasn't here to defend her role either. So what is the structure of DNA? What did they come up with? So DNA really is built primarily on 4 bases: adenine, thymine, guanine and cytosine. A, T, C, G. adenine like thymine, guanine like cytosine, they pair. And they give us a structure that looks like that. It's like a ladder. Only when the structure was first discovered, and in fact the way we understand it today, is it's actually a helix. So it's a helix that twists on itself, known as a double helices, as can be seen there. So in our normal human, what does this look like? Well, this is a normal human karyotype. So these are cells that have actually been broken open, and the DNA material is condensed into chromosomes, as is shown here. You can also see some cells there where the chromosomes have not been released. So if we look down a microscope, this is sort of the thing we see at relatively high magnification. Now, there are people who are much cleverer than me who know how to do this, which is they take that mass of lines, if you like, and can actually say what chromosome is what. So we know, and looking at that picture, you can see that the human genome is very ordered. We have 2 chromosome 1s, 2 chromosome 2s, 2 chromosome 3s, as you can see. So we have 22 autosomes and the sex chromosomes, X or Y, as can be shown there. Very ordered, very neat, very tidy when we get them lined up like that. I like this style because I can't tell what's a chromosome 1. I can probably tell what a chromosome 1 from a chromosome 22 is, but I'm not very good at telling what's a 1 to a 2 to a 3. So if we have a reasonable amount of money and want to look quickly at what the chromosomes are, we can do this and we can colour them, we can paint them. And then it's really simple for people to line them up because you're only matching colours then. So this is what the normal human karyotype looks like. It's relatively ordered. We have 2 pairs of each chromosome. We each have, if we're male, we have XY. If we're female, we have XX chromosomes. All very nicely organised. I keep doing that. Right. So it's great to have that molecular material or genetic material, but it really needs to be able to replicate. And this again is a process that's very ordered. So to replicate your DNA so that you can actually generate an identical cell, the cell needs to actually generate a complementary strand. This occurs every time your cell divides. So a cell divides, it gives rise to 2 daughter cells, and to do that, the strands separate, as is shown here. They rebuild themselves, as is also shown here, so you can see there the strand separation, some other bases coming back in, and the strands reform again. This happens millions of times a day in every cell of your body. And it really is, this is the first step of a new life, new cells, new division. It precedes something called mitosis, or cell division. So this is, as I said, the formation of 2 new cells. So we see the DNA replicates, it reproduces itself, it then, if you like, pulls apart, and we have 2 cells with identical. So these cells are identical copies of the parental cell. If you like a prettier picture, this is a little bit what it looks like. The cells line up on the metaphase plate, and the nuclei they recondense, the nuclei pulls together. So this is a normal process. Your skin goes through it, your hair goes through it, every cell in your body reproduces itself as frequently as it needs to. So I think we'd all recognise that the human body is very clever. There are times when it goes wrong. And there are several points that this can go wrong. So your A's and G's and C's and T's may not line up. So your A may not want to line up with your T, it might line up with a G. This is called a point mutation. It's actually an abnormality that occurs within a cell at the time of division. Certainly, as you saw, those chromosomes can be long, very spindly looking things. And when they line up at the metaphase plate, particularly during cell division, sometimes they get tangled and sometimes they break. And sometimes where they break, they rejoin somewhere else. And if that occurs, that's a chromosomal translocation, and that's an abnormality as well. And these are important concepts to understand as I move into talking about cancer. So we should all be a little bit worried about point mutations and translocations. They both result in abnormal proteins. They will, if left within your system, result in an abnormality mostly. And scarily, it's estimated that these occur 1 million times a day. So in your body, something goes wrong 1 million times a day. We're all still standing, we're all talking. Everything seems pretty normal on the surface. And that's because most of you wouldn't realise that the body's actually even cleverer than what we give it credit for. We have inbuilt mechanisms to get rid of those cells. So if we have a cell that has a point mutation or a cell that has a translocation, there are enzymes, for want of a better word, that proofread our DNA. So before it actually forms into another cell, before it then can reproduce itself, these enzymes come along and they say, over and done with, you don't need to be replicated anymore. And that cell either goes into senescence, which means it remains dormant, or it goes into programmed cell death, which means it actively is killed. So very clever. We have a very clever system for proofreading, making sure everything that is actually continues throughout your body is as normal as it can possibly be. What about in the abnormal situation? In a cancerous situation, these genes are allowed to go on. We get replication of an abnormal genome, and this really is the basis and the building blocks of the disease entity that we know of as cancer. The other thing that's important with proofreading is that the ability to proofread decreases with age. The incidence of cancer also increases with age. So this tells you that as you get older, with each division, your body just gets a little bit tired. You know, it's doing a lot of work every day. And the incidence, the ability of your proofreading machinery to actually grab that cell and throw it away becomes less and less. So it requires a little bit more help sometimes to actually do that, and as a result, the incidence of cancer increases with age. So let's go back to this slide. Let's talk about the new normal human karyotype, and we can see this is what it looks like. I've said to you it's ordered. It contains X number of autosomes and our sex chromosomes. What about a cancer karyotype? It looks a bit like this. This is actually breast cancer. Still kind of looks a bit ordered, but there's a lot of it. So we can see here that we have 4 copies of chromosome 2, at least. We've got at least 5 copies of chromosome 5, and generally we have a whole heap of stuff that we've got no idea. Even the experts have no idea what it is. So this is disordered chromosomes. A lot of them, a lot of changes. If you look at them the way I like looking at them, which is when they're coloured, you can see that this doesn't look nearly as clear as what it did when I showed you the previous photo. And I've just highlighted here some of the changes. So what we can see in a cancerous genome is that, for example, now I have to line it up, we see a little bit of chromosome 1 on chromosome 3. We can see that we're seeing a bit of chromosome 15 moving on to chromosome 11. We've got chromosome 2 on chromosome 19, clearly in places that shouldn't be. What that does is actually cause gene changes. It maybe if we're lucky, cause a stop codon and stop this gene from going on and being transcribed. But mostly what it does is lead to abnormal proteins. This is a seriously abnormal karyotype. This is a seriously abnormal cell. It's not going to produce any normal or very few normal proteins that we require. So this is great. We know that we can identify these chromosomes and we can identify these abnormalities. This is really no more than a screening tool. At any one of those point mutations, there's probably 5, 6, 7 or more thousand genes sitting there. So it doesn't actually help us identify it. It says there's an abnormality on that chromosome, but it doesn't really help us identify it. And if we can't identify what that abnormality is, then we probably can't do much about it. I'll learn eventually which computer to touch. So if cytogenetics is a screening program, how do we look deeper? So we didn't really have much of an ability. If we knew what genes were, what genes of interest may be around, then we would certainly be able to look at individual genes. But to look at the entire human genome really wasn't possible. So there were certain genes that were associated with different cancerous states, that we had certain probes that we could look at, but this was very directed. We knew very little until incredibly recently about what the whole human genome looked like. So the Human Genome Project, and excitingly today, it still remains the biggest international collaborative science initiative. So this was a cross-continent initiative. The remit of this group was to sequence the human genome in its entirety. There was, and I'm not actually sure whether it came in on budget, a $3 billion project. It was largely initiated by the National Institutes of Health, but it certainly included large groups from Europe, Wellcome Sanger Trust, the UK, China, Australia and others are really sort of put in there in grey because I don't actually sure what Australia did in this, but they are listed in the program. But certainly the driving force was the UK, Europe and the National Institutes of Health. So the idea was we needed to know what the human genome looked like from beginning to end. That is every piece of DNA. What it looks like, what are the genes, what are the structures of those genes. So the group, the scientists who were doing this, were given a timeline of 15 years. It was begun in 1990 and it was completed in 2003, 2 years before it was due to be completed. It was actually went deeper than they thought it would as well. So this is an increasing technology that caused this to go quite rapidly. What it does, in very simple terms for us, is it gives us a reference sequence. This is the normal human genome. It gives us a reference sequence to go back to. So when we sequence something, we can go back to RefSeq and say, look at this, let's line it all up. It doesn't line up. Or here's your abnormality. Here's the gene that's gone wrong. What can we do about that gene? So it's incredibly important for the face of diagnostic technologies that we use now and into the future, and for treatment modalities that we use now and into the future. So just as an aside, I have one of these in my lab. This is a HiSeq 2500. It is a gene sequencer. It does whole genome sequencing. It does what the Human Genome Project set out to do. And obviously companies like Illumina have made a fortune from the Human Genome Project. They make good products and many people buy them. What's important is that this can sequence a human genome to probably 200 to 300, I can go deeper if I want to, depth. So that's 200 or 300 times over the genome. And it can do it in 5 to 6 days and it will cost me about $1,500. So this is technology that is just booming. We can now take not only cancer, but microbiology and a variety of other things. We can actually sequence the bugs that we need to be looking at, the genomes that we need to be looking at very rapidly and get a lot of information. So this really is, we're just coming to the cusp of this boom. And to be quite truthful, and I think Alistair would agree, many of us are really struggling to actually keep up with it because these things generate huge amounts of data that we need to now, I don't only need to have scientists in my lab, I need to have bioinformaticians, mathematicians, people who can decode what I call that black box of data that comes out, because I can't. So this is, we're really at a very exciting precipice, if you like, of the way that we're going to approach diagnostics and cancer from here on in. So let's touch a little bit on what cancer is. It's a group of diseases which involves abnormal cells, and I've shown you some of the reasons for that. These cells have the potential to invade or spread to other parts of the body, and of course this is taken from a very reputable source called Wiki, or Wikipedia. It's a pretty good definition. It is in fact a pretty horrible diagnosis for anyone. However trivial, however early, for a cancer patient to be told, for an individual to be told they have cancer, even if only for a short time, things are very chaotic. One of the things that most cancer patients come up with is, what are the choices and what road should I take? And I guess I want to spend a little bit of time now talking about what the choices are and what the new choices may be, and how we're using the new technology to change the choices that cancer sufferers or newly diagnosed cancer patients actually face. So before we could understand more about cancer, we knew it was an uncontrolled growth. We didn't necessarily know what genes caused it. We knew the DNA was a problem, that there were a variety of different genes that would be involved. But really what we needed to do was take a wholesale look at someone with cancer and treat them as rigorously and hard as they could possibly be treated. So these modalities would be things like chemotherapy, which stop the cancer cells dividing. The only problem with standard chemotherapy is it stops every cell dividing. So it doesn't only affect the cancer cells, it affects your normal cells as well. The importance of using chemotherapy in this setting is that cancer cells divide much more rapidly than what normal cells divide. So they tend to be killed at a much greater rate. It's an effective therapy, but it has amazing toxic side effects, and it's not effective in all diseases. Radiotherapy, another modality where patients are really treated in a very directed fashion, and of course surgery. So these might be the choices that even today patients are given. Chemotherapy, radiotherapy, surgery. It's still really the three choices in the majority of cancers. But if we think about chemotherapy, which I mentioned, versus what could be possible if we know a little bit more about the cancer, and that's targeted or rational therapy. So therapy that actually directly targets the genes that cause the disease. Let's do a comparison. This is kind of a little bit what chemotherapy is. So we have what hopefully to start with is a little target, and we have a hell of a big arrow going towards it. So this is, we're really not only, we're using, if you like, a big bullet at a very small target. The other alternative is that we're using multiple chemotherapy modalities and we're hitting lots of other things. We might actually be hitting our target, but the off-target side effects might be unbearable and dangerous. What about if we compare that to rational therapy or targeted therapy? If we know what the target is and we have the drugs available, we can target it. They're all criteria that everything needs to line up. But a lot of what we do, my lab is very translational, it's considered a translational research laboratory. And most of what we do is explore and understand the underlying disease biology of these cancers, including, and actually very much at the forefront now, genetics or genomics. And I need to add that I'm actually not classically trained in genetics or genomics. We had to learn very quickly. Using this sort of approach, we can define targetable lesions. We can test drug sensitivity in vitro. So we could, and we do, use genetics to actually define the underlying cause of the leukemia. We can then, in the laboratory, in a test tube, very simply put, if you like, expose these patients' cells to drugs to see whether they're sensitive or not. And from this, we can build the future clinical trials to try these drugs in a larger cohort of patients. So this is sort of the future of therapies in the cancer setting. I can talk a lot about it, but have I got any evidence that this actually works? So we need to take a step back to the early 1990s, and this is really the first era of targeted therapies. So this is, as you would be aware, before the Human Genome Project was completed, in fact, when it was just started. But the first ever cancer, leukemia, that was caused by a known genetic abnormality with chronic myeloid leukemia. And this is something that I've worked on for the last 20 years. So these are survival curves. The probability of survival is on the y-axis, and the years from diagnosis is on the x. And we can see incremental increases in survival over time, starting back in 1983. Is that what that says? With simple chemotherapy agents like busulfan. But really the biggest jump that we saw was a 30% increase in survival in the last 10 years. And why was that? That was because we understood what caused this cancer, leukemia, and we could target it. So what do we know about CML? This is chromosome 9 and chromosome 22. Chronic myeloid leukemia is caused by this. It is a reciprocal translocation that takes a bit of chromosome 9 and puts it onto chromosome 22, and takes a bit of chromosome 22 and puts it back onto chromosome 9. This happens to bring together some crucial genes. This chromosome, by the way, is called the Philadelphia chromosome. That was because it was identified in Philadelphia. This brings together two genes, the BCR gene and the ABL1 gene. It transcribes into a protein called BCR-ABL, which is, if you like, a powerhouse. What BCR-ABL does is make this cell put the accelerator on and take the brake away. These cells divide. I can show you, in fact, this is what the blood of someone with chronic myeloid leukemia at diagnosis looks like. Incredibly crowded, probably doesn't mean that much to you. This is what normal looks like. So these patients with chronic myeloid leukemia produce huge numbers of cells. They're relatively normal cells, actually, a little bit immature, but relatively normal cells. The reason it produces these cells is because BCR-ABL is an activated gene. ABL is in itself an oncogene, so it's cancer-causing. Bring BCR in close proximity with it, it switches it on and says, you're not going to stop now. It removes the brake mechanism on that cell and causes these cells to divide and replicate. These patients often present with an inability to see because their blood is so thick with white cells. It was, and probably actually still is, one of the most well-recognised treatment and developments in Western society. It made the front cover of Time magazine. I'm not sure, the Human Genome Project certainly made the front cover of Time magazine. I'm not sure whether any therapy since has. But this was the, as said in Time magazine here, the ammunition against cancer. These are the magic bullets. What does Gleevec do, or imatinib, Gleevec as it's known, it really is a drug that fits within the active domain of BCR-ABL and stops it working. It gives it a break. So structural chemists took BCR-ABL, they designed a drug to fit exactly within the binding pocket and shut it down. The cells, the leukemic cells, actually become dependent on BCR-ABL, and as soon as you shut that protein down, they die. We thought, and I had no involvement in the development of this drug, but I worked very early with it, in the very early days with it, and we thought actually when we shut the protein down, the cells would actually revert to normal and then become abnormal again. The truth is it shuts them down, they die. Remarkable improvement. You saw those survival curves. Remarkable improvement. Why? Because we actually understood the causative lesion, the causative genetic lesion, and we could target it. And we know that this is the first successful treatment, and because it's been so successful, in fact, there are now many other drugs within the same class. It's so successful that we now, after 10 years of treatment, have patients that we take off therapy. And for reasons that we still don't understand, but it's a big area of research in my lab, approximately half of those patients, after they've been in complete remission on Gleevec, we can take off and they don't relapse. It's cure, but I'm a bit scared to say cure at the moment. But certainly these patients are doing incredibly well. So this is an incredibly effective drug that targets the causative agent. Other area I work on is acute lymphoblastic leukemia. This is a neoplasm of lymphoid progenitor cells. It really causes a maturation arrest. These cells are not functional. They don't do anything that we normally expect a lymphoid cell to do within your body. They provide you no immune help, assistance if you like. These are nasty, nasty leukemic cells that are floating around, and there's a picture of them there in the centre. It is the commonest childhood tumour, and there is a 80% disease-free survival. So that's great. 80% of our kids are cured if they have acute lymphoblastic leukemia. Depends whether you're a cup half empty or cup 3/4 empty, cup whatever the rest is, quarter full sort of person. But that means 20% of our kids die of our children diagnosed with ALL die of their disease. If this is the commonest childhood tumour, then it doesn't take much to realise that this is probably the leading cause of death. So ALL remains the leading cause of non-traumatic death in the youngest members of our community. This is a disease we need to do something about. If you happen to be my age and diagnosed with ALL, your outcome is incredibly dire. In fact, some of these patients don't make it past the ER. So this is a nasty, nasty disease. Chronic myeloid leukemia, we've been very clever with it. It's a more indolent disease. It gets nasty, but this is at diagnosis a nasty disease. So you remember I showed you the karyotype, the cytogenetics of the leukemias? This is what the cytogenetic classification of acute lymphoblastic leukemia looked like in the year 2000. It actually looked identical today. So we'd gone as deep as we possibly could with cytogenetics. We knew as much about ALL as we could possibly know using basic cytogenetics techniques. Using the Human Genome Project, we can now go further. So a lot of these subtypes, and this is all the pie diagram shows you various subtypes that could be identified cytogenetically or karyotypically. There were, however, 27%, 25% of patients with acute lymphoblastic leukemia for which we knew nothing about, but we knew they were really bad players. These patients didn't do too well, and they needed to be defined. So the Human Genome Project allowed us to do that. And I won't go into the details of this because this slide gets a little scary. But I really want you to take home, the take-home message from this slide is this is 15 patients diagnosed with acute lymphoblastic leukemia. Using routine cytogenetics, we can see that 3 of them, the 3 there with IGH CRLFL2, so these are 2 genes that are fused, we can say, yep, this is what these patients have got. This is what's driving their disease. With the sequencing of the human genome now, what we can look a little bit deeper. And you can, even if you're not scientific in the audience, you can see that going from one column to the other looks a heck of a lot more complex. That's because we can look a bit deeper and we can discover that there's other things within these genomes. When we look deeper still, they're complex. So remembering that cytogenetics couldn't pick up any of this, but if we look at the genetic level, these patients have complex abnormalities. There are multiple genes that are altered in these, this is kids' work, children's work. There are multiple genes, many of them are recurrent, and most of them actually just are responsible for stopping those cells from maturing and causing them to become cancerous. So this is critically important for this group of patients that we understand what's driving their disease. Again, a little bit of a scary slide, but what I really want to show you here, I don't know whether this is going to work, no it's not. If we look, you can see the lines, the EBF1 and PDGFR beta. This is actually deep sequencing. So this is us lining up the sequences. We sequence someone's genome, and that top line you see there is our RefSeq. This is what we get from the Human Genome Project. And using this and lining up the sequencing of someone's tumorous cells, we can actually tell what gene is coming into proximity with another gene. We can then, and I really just want to show you a snapshot of this, we can then take those cells, put them into culture and ask, does that lesion actually cause the cancer? And in this case, as highlighted there, a particular fusion, when we put it into culture, goes crazy. and that is actually the cancer-causing agent, or the cancer-causing fusion. What's really good is that then we can use different drugs, in this case dasatinib, it's a tyrosine kinase inhibitor. It actually works in chronic myeloid leukemia. We now know that it actually works on a subset of acute lymphoblastic leukemia. This is cell death, so what you really want is your curves to be coming down, as you can see here, in the solid lines, not the dotted lines, and we can tell very rapidly. We can see what the translocations are, we can see whether they're transforming, and we can see what we can treat them with. So we set up all of these things when a patient's diagnosed. So let's talk about a good news story. This is a young lad. I'm not breaching any problem. I don't have any problem showing his photo. He loves having his photo taken. He's a 10-year-old boy at the time of diagnosis. He's now 13. He had refractory acute lymphoblastic leukemia. In fact, at day 29, he was facing a very dire outcome. He was palliated at the age of 10. Cytogenetics indicated that there were some alterations, and he was deep sequenced and found to have an EBF1 PDGFR beta fusion. So two aggressive genes really, coming together that were actually driving this disease. This patient was palliated. This is a 10-year-old. He was going to die. So because in that scenario you can try experimental medicine, he was put on imatinib. It's a safe drug, we know, but it's not usually used in this setting. Within a week he responded. He had a morphological remission. Within 2 weeks his disease was almost undetectable, and this slide's now a little bit old, but he's now 3 years out and he remains on treatment. The tricky bit, he remains on treatment but in remission. So there is no doubt that this child's life has actually been saved by being able to use a targeted therapy. He is the poster child. This is a rather busy and actually albeit ugly slide, but this is looking at the children that we're working with at the moment in acute lymphoblastic leukemia. I don't want, there'll be no tests on this afterwards. I don't want you to try and understand nor read, actually, it's probably hard to read from the back of the room, what these genes are, but I really just put it up there to show the complexity. A lot of these fusions, the majority of the things that I've put on that screen actually cause the disease. And using deep sequencing we can actually understand them, and we can hopefully target them. So that's the sort of nuts and bolts of CML and ALL. I just want to spend the last 3 slides probably talking about randomised control trials and the new face of how we're going to treat patients now that we've got all these other techniques available to us. So what happens in a randomised control trial? Some of you may have been involved with them. So really it takes a group of patients and it has a treatment group and a control group. This could be for a antibiotic therapy, it could be for a chemotherapy regime. Really what happens is that as a patient's diagnosed, they're randomised, and they can be randomised to A or B. It's usually, but not always, blinded, so the patient actually doesn't know what they're on, or at least the clinician doesn't always know what they're on. Sometimes it doesn't mean anything to the patient anyway. So the treatment group is compared to the control group. In cancer therapy, this tends to be the control group would be current standard of care versus treatment group, which would be current standard of care plus or new standard of care. A direct comparison. The funding bodies require us at this stage to still do these sort of trials, to actually compare next best with previous best or next possible best. Results are compared, and this provides an indication. You saw in that last slide, the reason I showed that rather ugly last slide with lots of detail on it was to show that we actually now know we've got many targets. So is a randomised, either single or multi-agent randomised controlled trial the best approach? We can't actually use imatinib on someone who doesn't have a fusion, or dasatinib, on someone who doesn't have a fusion that will actually be treated by that drug. So is using a randomised trial ethical and in fact possible? We know that in the first ever trial of imatinib in chronic myeloid leukemia, that trial was ceased, incredibly successful trial. The trial was ceased because the new drug was so much better than the standard of care that it was unethical to leave patients on standard of care, and they're all allowed to switch to the new therapy. So we really, in the future, need to be looking towards a personalised approach based on causative lesions. And as you can see here, rather flippant, but it's all about the genome. We have something that's just for you. This is a real change in mindset. We haven't thought this way before about the treatments. I've just come back from the States, actually, where we were talking about these sorts of trials, and this is looking at what we call either basket or umbrella trials. So randomised controlled trials is one thing when we talk about one drug, one genetic alteration, but many organs. So that means that if you have leukemia, you might be sitting in the clinic alongside of someone who has lung cancer, alongside of someone who has a kidney tumour, because they're all driven by the same lesion. So they can all be treated together, even though they're not the same disease. The alternative approach is to look at multiple drugs in multiple genetic alterations, all in the same organ. So you can see we're really starting to sort of have to think outside the square as to how we use the new information as to how we treat the patients. But there is no doubt that the new paradigm will require, new treatment paradigm will require multidisciplinary sequencing tumour boards. I get sequences put before me almost every day. Some of them I have no idea what it means. We have genes that are coming through that we don't know if they're causative. We can clone them and put them into cells and ask that question. But we really need to bring together a multidisciplinary team. It won't just be your oncologist. It will be your scientist, your oncologist, your bioinformatics specialist, your basic pathology who have always been involved, your clinical geneticist, and importantly, and I won't delve into this in the, because time's getting now short, your bioethicist, the person who talks to you about what your rights are here and who decides how ethical it is to move certain processes forward. So incredibly exciting, but an incredible paradigm shift and an incredible change. Did it again. So what are the obstacles? If your DNA test shows that you're predisposed to sue doctors, it's probably going to be a problem for you. And we do need to consider how we use this information. There are people who would sequence every child at birth. Should we do that? That's an ethical conversation for another talk, a very long talk. But certainly we could. We could sequence everyone when they're born. The cost of sequencing is going down. I think the thing, and I don't often quote Muhammad Ali, but the thing that I think we need to understand is that there is a mountain ahead. There is a lot for us to understand. But in fact, we've climbed one hell of a mountain over the last 10 years and changed the face of our diagnosis, or provided the mechanism to change the face of our diagnosis. The biggest issue now is going to be the pebbles in those shoes. Getting our drug companies to talk to each other, it's a bit of a pebble. Getting ethics to agree with that these are the approaches, that's probably a bit of a pebble. But we have made incredible inroads. This is not the royal me or we. This area has made incredible inroads, and we're really now at a critical stage. So we've climbed that mountain, and yeah, we've got a lot of pebbles, but I think we now have the tools to get through this. Thank you.